This video we are looking at the steps which are involved in the TCA cycle. So to start off with a few questions, explain the process of the conversion of citrate to the isocitrate. Next, what happens during the conversion of the keto glutarate to the succinyl CoA. Next is explain the step in case of the succinate to the fumarate conversion. So we look at all the answers in this video and learn a lot more. So firstly, the pyruvate, it is converted to the acetyl CoA. Then it goes to the citrate. Then the citrate is converted to the isocitrate to the alpha keta glutarate, then to the succinyl CoA. Next, the succinyl CoA is converted to the succinate. Succinate is converted to the fumarate. Fumarate is converted to the malate. Malate is converted to the oxaloacetate, and then it again goes to the citrate. So this is the outline what exactly happens. Now we are going to break through the enzymes which are involved in this process. So firstly, the pyruvate is converted to the acetyl CoA. So the enzyme which is involved here is nothing but the pyruvate dehydrogenase. So here what happens is the CoA plus the NAD, it is converted to the CO2 plus the NADH plus the H plus. Next, we see that the acetyl CoA, it adds on to the citrate. So here what happens is there is addition of water, and what is released is the release of the CoA. So the enzyme involved in this process is the citrate synthase. Next, we look at the conversion of citrate to the isocitrate. So the enzyme involved here is nothing but the aconitase. Next, what we see is the isocitrate being converted to the alpha keto glutarate. So here the NAD plus is converted to the NADH H plus. So what is the enzyme involved here? Enzyme involved here is nothing but the isocitrate dehydrogenase. The next step we see is the alpha keto glutarate being converted to the succinyl CoA. So the enzymes here, what happens is the NAD plus is converted to the NADH and H plus CO2. So what is the enzyme which is involved in this? It is nothing but, as the name suggests, is alpha keto glutarate dehydrogenase. So here we saw that it is nothing but the NAD plus the CoA SH. Then moving on to the next step, that is what happens in case of after the succinyl CoA. So we get succinyl CoA. So after the succinyl CoA, there is the succinate. So the succinyl CoA, the enzyme here is nothing but the succinyl CoA synthetase. So what happens here is the GDP plus, and this is converted to the CoA SH plus the GTP. So here it is GDP, here it is GTP. So after this, what we got was the succinate. So after the succinate, there is formation of the fumarate. So this fumarate, succinate to the fumarate, is in presence of the enzyme. What is the enzyme? It is nothing but the succinate dehydrogenase. So what happens here is that the Q is converted to the QH2. Next, what happens after the fumarate? So after the fumarate, it is converted to the malate. So fumarate to the malate, enzyme here is nothing but the fumarase, and this is in presence of, there is presence of the water. Next what we see is the malate is converted to the oxaloacetate. So what goes in this process? So the enzyme here is nothing but the malate dehydrogenase. And what happens here is that the NAD plus is converted to the NADH plus H plus. Then moving on, there was another step where the pyruvate, it started with the pyruvate. So the pyruvate, it directly goes on to the oxaloacetate. So in this special step, what happens is, this is in presence of the PC, that is the pyruvate carboxylase. So the enzymes here is nothing but this HCO3 minus plus the ATP. So this step is converted to the ADP plus the PI. So now going into all the individual steps. So step 1. In case of step 1, there is the acetyl-CoA plus the oxaloacetate. So this gives rise to the citrate. So what we see here is nothing but 2C plus 4C. What we get is 6C. So the enzyme here is nothing but the citrate synthase. Then what is the speciality here is that it is irreversible, as well as it is a exothermic reaction. Next we look at the step 2. So in case of step 2, the citrate is converted to the cis aconate and then to the isocitrate. So what about the carbon? So citrate is 6C and aconate is also 6C and isocitrate is also 6. So looking at the enzyme which is involved here. So the enzyme here is nothing but the aconitase. It is the enzyme which is involved. So looking at the cofactor is nothing but iron. Then looking at the reaction it occurs in 2 places. That is 2 steps. So the first step is the dehydration. And the second step is nothing but there is rehydration. So how about being inhibited? So it is inhibited by the fluoroacetate. It is nothing but the NC, that is non-competitive. Then we move on to the step 3. So the step 3 is nothing but the isocitrate is being converted to the oxalosuccinate to the alpha ketoglutarate. So the isocitrate is nothing but the 6C, oxaloacetate also the 6C, but the alpha ketoglutarate is the 5C. So then looking at the enzyme which is involved in this step. So the enzyme here is nothing but the isocitrate dehydrogenase. It is ICD. So what is a cofactor? The cofactor here is nothing but the magnesium as well as the manganese. So what about the reaction? The reaction is nothing but oxidative decarboxylation. What is the intermediate? Intermediate is nothing but oxalosuccinate, which is nothing but unstable. So what does this release? So release here is of 1 NADH as well as the CO2. Then we move on to the step 4. So in case of step 4, we see that alpha ketoglutarate is converted to the succinyl CoA. So alpha ketoglutarate is nothing but the 5C is being converted to 4C. So what is the enzyme? Enzyme here is nothing but the alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase. So it is nothing but a multi-enzyme complex. And the reaction? Reaction is nothing but it is an oxidative decarboxylation. And what is it inhibited by? The inhibition is by the arsenite. It is non-competitive. And physiologically it is a unidirectional step. And what is released in this step is that there's release of 1 NADPH as well as the CO2. Then we move on to the step 5. So in case of step 5, what we see is that there is a succinyl CoA being converted to the succinate. So here what we see is 4C is being converted to 4C, and the enzyme here is the succinate thiokinase. And what about the cofactor? The cofactor is nothing but magnesium. And what does it release? There is release of the 1 ATP as well as GTP. Then we look at the substrate level. There is a substrate level phosphorylation as well. Next, moving on to the step 7. So in case of step 7, there is a conversion of fumarate to the malate. Here it is 4C and here also it is 4C. So what about the enzymes involved in fumarate to the malate? So the enzyme is nothing but the fumarate hydratase, and then the reaction is nothing but there is hydration. Now moving on to the last step, that is step 8. So the malate here is converted to the oxaloacetate, and looking at the carbons, it is a 4 carbon and oxaloacetate is nothing but a 4 carbon. So what about the enzyme? The enzyme here is nothing but the malate dehydrogenase. And what does it release? The release is of NADPH, and other thing is the oxaloacetate is regenerated. Now to answer our questions. So the first question we asked was, what happens in case of the conversion of the isocitrate to the alpha ketoglutarate? And the next question was, first question we saw is that the isocitrate being converted to the alpha ketoglutarate. Now to answer the questions, the first question we asked was to explain the conversion of the citrate to the isocitrate. So firstly the citrate, that is, it is a 6 carbon, it is converted to the cis aconite. This is again 6C. Then it is converted to the isocitrate. It is nothing but again the 6C. So what is the enzyme which is involved here? So the enzyme here is nothing but the aconitase. So what about the cofactor? Cofactor is nothing but the iron. And other thing to note is that the reaction, it takes place in 2 steps. So the first step, it involves the dehydration. The second step, it involves the rehydration. And it is inhibited. The inhibition is by the fluoroacetate, and this is by the non-competitive. Next is the conversion of the alpha ketoglutarate to the succinyl-CoA. This was the next question which was asked. So here it is the 5C being converted to the 4C. And looking at the enzyme. The enzyme here is nothing but the alpha ketoglutarate dehydrogenase. And it is nothing but a multi-enzyme complex. Then looking at the reaction. The reaction, it is oxidative decarboxylation. And there is inhibition. The inhibition is by the arsenite. It is nothing but it's non-competitive. Next is physiologically, it is a unidirectional step. Okay. Then there is release. There is release of 1-NADPH as well as the carbon dioxide. So this was the second question. Then moving on to the third question and the answer. The third question we asked was, in case of the conversion of the succinate to the fumarate, what are the steps involved? So in case of succinate to fumarate, succinate is 4 carbon, fumarate is 4 carbon. So the enzymes here is nothing but there is succinate dehydrogenase. So here it is nothing but the part of the second complex ETC. So the cofactor here is nothing but there is FAD as well as the FES protein. Then the only enzyme of the TCA cycle attached to the inner, it is the only enzyme of the TCA cycle which is attached to the inner mitochondrial membrane. It is inhibited by the malonate and it is competitive. And there is release of the FADH2. Hello everyone, this is Dr. Hub. Now let me tell you our story. Running with us, we are sure you will reach your destination with ease.